All right. Hey guys, Dr. Eisenbach, chapter 21, Microevolution. So, in the last chapter, we kind of got an introduction to evolution and we looked at some of the historical context. And we looked at, what did we look at? We looked at, kind of brought it up to date or bridged the gap between Darwin and modern theory. And for this chapter and the next couple, we're going to be looking at mechanisms of evolution. In this chapter, we're looking at microevolution, and then we're going to look at speciation and macroevolution. So for today, let's just begin at the beginning of the chapter by talking about some topics that kind of often come up when we talk about evolution. One is resistance. So bacteria are really good, as we talked about earlier this semester, at swapping genes, swapping plasmids, creating genetic variation by just kind of loosely trading and swapping sections of DNA. And what we found is that when we apply an antibiotic to a bacteria population, that over time it can develop resistance. And I'm sure you're aware of this, but the book talks about how when penicillin was first invented, it did a really good job um, of suppressing bacterial infections. But over time, penicillin became less effective because bacteria were gaining resistance. And so resistance is just an example of microevolution in process. In other words, those bacteria display variation, they have diversity, and the penicillin wipes out and kills the members of a bacteria population that do not have resistance to it. However, the bacteria that do have resistance to the penicillin will survive and reproduce, and so over time your population of bacteria shifts from one that is mostly susceptible to the antibiotic to becoming a population that is mostly resistant to the antibiotic. So that's resistance. That's also an example of microevolution. So microevolution is simply a heritable change in the genetics of a population. So in other words, it's evolution, but it's on a small scale. So when we talk about microevolution, micro meaning small, we're usually talking about evolution that's happening in relatively short time frames, or it's happening within a population to create two subspecies, or maybe to create a species. So microevolution is often at the family level or below. When we talk about microevolution, it's important to understand that we're dealing with populations that are evolving. So it's not individuals that are evolving, it's populations. So what's a population? A population is all the individuals of a single species in the same place at the same time. So campus, if we were there, has a population of gray squirrels. So we could say all of the gray squirrels that are on Milligan's campus right now in March 2020 is a population. And if we were to study that population of gray squirrels over time, we could see that it might shift. So they might get a little bit, their color might change a little bit over time, or their behaviors. And so typically when we're studying evolution, we're studying populations. We're not looking at an individual squirrel and expecting it to evolve. We're looking at a population and we're expecting it to evolve. Okay, so how do they evolve? How does a population evolve? Well, there's multiple mechanisms that change populations that cause them to evolve. And we talked about the one that Darwin came up with, that Darwin discovered in the last chapter, and that's natural selection. And you're going to see in this chapter that there's several mechanisms that cause evolution to occur. So we'll come back to that point later in the lecture. So we know that populations have natural variation. So meiosis, if you recall, creates genetic diversity in the gametes that are produced. And so the whole point of sexual reproduction is to generate organisms that have diversity, or a new generation that has a diversity with lots of different alleles represented. And so phenotypic variation is simply differences in appearance or function that are passed from generation to generation. So in other words, I hope you know what a phenotype is. It's when you're looking at an organism, it's what it looks like. And populations have phenotypic variation. So if you look at a population, if you're looking at a population of gray squirrels, you would see that they vary. They have phenotypic variation. Some of the squirrels might be more gray, some might be more brown, some might be larger, some might be smaller. If you were to do some dissections, you might see that their internal anatomy differed a little bit. And so those are all examples of phenotypic variation. So the individuals of a population aren't clones of each other. They each have a unique genetic code because of the process of meiosis and sexual reproduction. In the picture, you can see examples of phenotypic variation. The top picture is European garden snails. The bottom is I thought it was Bahamian, but Bahaman, I don't even know what that is. Bahaman land snails. And in the top picture, you can see all these different color patterns. All right, so this is an example, a picture of phenotypic variation. All right, if you look at the land snails in the bottom picture, you see that they look pretty similar. So they all have phenotypic variation, but it's not as evident. In other words, it's not showing up as obviously in their shells. They look very similar. But trust me, they have phenotypic variation. So sometimes it's going to be more obvious to the observer, and other times we have to go looking for it. On that note, let's talk about some categories of variation. So quantitative and qualitative variation. So quantitative variation is when individuals differ in small incremental ways. Okay, so if we were to take everyone in class and if we were to weigh everybody, we would see that probably no one weighs the exact same amount. So the weight varies almost continuously from the lightest person in the class to the heaviest. Or we could do the same thing with height. And so we could graph the height or the weight of people in our class, and it would probably create a nice bell curve. So most individuals would be at height that's considered an average height or mean height. They would cluster around the mean, and a few of us would be tall and a few of us would be short. And so in the picture on the right, in the upper portion, you can see that this would be a hypothetical bell-shaped curve representing the quantitative variation of height or weight in the population that is our class. Compare that with qualitative variation. In qualitative variation, the traits exist in two or more discrete states, and there aren't intermediates. Okay, so in this example, look at the picture of the geese. These are snow geese. And so the snow geese are either blue or white. That's according to the book. It doesn't look blue to me. It looks brown and white, but whatever. That's what the book said. So the point is, in qualitative variation, you don't get a bell-shaped curve of coloring patterns in the geese. They exist in discrete colors. They're either blue geese or they're white geese. So this is an example of qualitative variation. When this happens, we often say that, or we can use another term called polymorphism. So polymorphism is simply the existence of discrete variants of a trait or a character. So we would say that these geese are displaying polymorphism. There's just multiple phenotypes, in this case 2, and they're discrete. They're exhibiting qualitative variation. So really polymorphism is something that happens when the variation is qualitative. All right, when we look at phenotypic variation, it's interesting to think about why do we have that? In other words, why do we have different heights or different weights in quantitative variation? Or if we're looking at qualitative variation, why do the geese exhibit 2 different polymorphisms? And one idea or one explanation of this is the interaction between genes and the environment. So in other words, some of the variation in a population is going to be from genes. So you're tall because you have tall genes, or you're short because you have short genes. But the environment also influences the expression of genes, and it influences the development of an organism. So maybe some of us are tall because our environment caused us to be tall. Maybe we're tall because we took our vitamins, or maybe we're short because we had a disease when we were a kid, or we didn't get essential minerals or nutrients. So, um, as an example of that, look at the picture of the hydrangeas here. We've got pink ones and blue ones in terms of their flowers. And so this is not a genetic difference. So in other words, the pink ones and the purple ones, the difference in flower color is not due to genetics, it's due to the environment. So with hydrangeas, if they're grown in acidic soil, they produce blue flowers. And if they're in neutral or basic soil, then they produce pink flowers. So you might be tempted to say, oh, these might be two different species. These polymorphisms are because of genes, but it turns out it's because of the influence on the environment, on the way that the genes are expressed. So it makes different nutrients available, it changes the physiology, and the result is different flower colors. This is important because only genetically based variation is subject to evolutionary change. So in other words, natural selection and the other mechanisms of evolution, um, they don't really cause evolution to happen if these phenotypic differences are due to the environment. So natural selection, for example, operates on genes. So it selects for genes or alleles that are beneficial and selects against genes or alleles that are not beneficial. And so we're really just interested in the genetic makeup or the phenotypic um, aspects of a population that are linked to genetics, because that's the data or that's the information that evolution is going to act on to create changes. So how do we know? How do we know if the differences in phenotypes are due to genes or the environment? And the book goes into a lot of detail with this, but since you don't have your books, I'll just summarize it. Basically, the idea is you have to do experimentation to figure out if the phenotypic differences are due to genes or the environment. And you do that by planting, planting some seeds. If we're dealing with plants, planting some seeds under conditions that are exactly the same. And if the conditions are the same, and then the phenotypes end up looking the same, then we could say, well, then the differences that we saw before were due to the environment. But if you plant the seeds in two different fields and the conditions are different, but they have the same genetic makeup or similar genetic makeup, then maybe the environment is the main cause, and it causes those plants to grow differently in the different fields. All right, so anyway, I don't know if that made any sense, but the point is um, we can do it through testing, and we have to do it through controlled experimentation. All right, where does this variation come from? So we saw those snails a few slides back that had all the different color patterns. And if it is genetic, and we're going to assume from this point on, we're really just talking about genetic sources of variation and not environmental. Where did this variation come from? So where did the alleles that cause, red striped patterns versus brown versus yellow versus pink versus purple, where did those different alleles come from? And the answer is it comes from mutation. So a mutation is simply any change in DNA. And there's different types of mutations, which you probably learned about in the first half of the semester. But there's different types of mutations. In each case, the mutation creates a change in the DNA, and the change in the DNA will cause a change in the RNA, and a change in the RNA can create a change in the amino acids that the DNA is coding for. And if you change the amino acids, then you're going to change the protein that's produced. If you change the protein that's produced, it's going to result in different characteristics, and that's the source of your variation. All right, so mutations change DNA, and that's where any changes or any variation begins in a population. Another way that genetic variation can occur or can appear is through not changing the DNA in the sense of changing an A to a T or a C to a G, but rather through rearranging the existing alleles. So you can see from the bullet points here that can happen in a couple different ways. So you can change the chromosome structure or number. So in other words, you might have 23 chromosomes, and then there's a mutation that causes that individual to have 24 chromosomes, so that's a source of variation. Last semester you learned about how in meiosis, prophase 1 has an event called crossing over, where homologous chromosomes line up and swap information. That's what's going on in this terrible picture on the right. And so crossing over is a source of genetic variation. It's where the genes get swapped around from different chromosomes, and that's what creates unique gametes at the end of the meiotic process. You can also have independent assortment of chromosomes. So this is happening in metaphase and into anaphase. So basically, after crossing over, these homologous chromosomes line up, and then they move to different parts of the cell, and then the cell splits into two, and then those two cells split into two more, creating 4 gametes. So the way that those chromosomes move in the cell creates genetic variation. And then you also can have random fertilizations. So random fertilizations is simply it's going to be random as far as which sperm fertilizes which egg. So eggs are produced through meiosis, and so one is just released during ovulation, in which there's millions of sperm that are racing towards this egg to fertilize it, and it's totally random which sperm gets there first. Not totally random, but it's pretty random which sperm is going to end up fertilizing the egg. All right, so these are just all sources of variation in a population. Populations have genetic variation, and we can look at this through a couple different ways. So one is biochemical polymorphisms, and so basically when scientists are studying species and trying to figure out how closely related they are or how different they are to determine their evolutionary relatedness, they can use techniques where they basically put genes or put sections of DNA onto some gel, and they zap it with electricity, and they can this electricity causes it to move through the gel, and they can separate different genes or different proteins based on the molecule's shape or mass or charge. And so we can figure out, using this technique, they can look at different polymorphisms of, say, a protein, and then they can figure out the genetic variation at the gene that codes for that protein. We can also do DNA sequencing. Well, let me back up. We can also compare genomes. So you can sequence the genome of one individual and compare it to another, or sequence the genome of one species and compare it to another. And you can basically look for similarities in the nucleotide sequences and look for differences. And if there's more differences, then they're less related. And if there's more differences, they're less related. And if there's fewer differences, then they're more closely related evolutionarily. SNPs or SNPs stand for single nucleotide polymorphisms. So if you look in the upper picture here, you can see that this individual, when you sequence a particular gene, is GCAA. This one is GCAG. This one is GCAT. And so it turns out that individuals have single nucleotide polymorphism. So we differ from each other just in some cases, in some genes, simply by having a different nucleotide base compared to others. And so this is used like if you're trying to determine if who the father is on a baby. If you're on what's that show? Maury Povich's show. You guys know what I'm talking about? Okay, whatever. You can see. So if someone has an A and their father has an A at this site, or if someone has an A but their father has a G at this site, then that's one data point that you can use to try to determine paternity. Another tool that we can use is STR variants. So STR stands for short tandem repeat. And so in our genome, there are sections where, for whatever reason, there's just these tandem repeats of a nucleotide. So tandem means back to back, and repeat means they're repeated. And so we individuals have different numbers of repeats. So if it, just look at the picture down here. So here we've got crime scene DNA, and they went to two different sites, and they're looking at the short tandem repeats at these two sites. So at site 1, the crime scene DNA is AGAT over and over. So this would be AGAT, 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 and at site 2 it would be GATA. So it would be GATA, 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 GATA over and over. All right, and then you take DNA from a suspect and you go to that same gene, the same site, and you look and see what their STRs are. And so this suspect has the same number of short tandem repeats. 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7. So it's not looking good. This person, they could be the criminal. However, when we go to site 2, we see that the number of short tandem repeats doesn't match. So the suspect has a few extra short tandem repeats. So this bodes well for them because this suggests that they are not criminal, and we can let them walk free, go free. So short tandem repeats and SNPs are just ways that we can kind of zoom in on genetic variation between individuals to try to distinguish their relatedness. All right, when we look at a population, populations have a gene pool. All right, and what that means is the gene pool is simply the sum of all the gene copies in all individuals. It's basically a summation of all the alleles that are available. All right, and so the population of gray squirrels on Milligan's campus, we could take DNA from each of them, a DNA sample, and we could determine the gene pool. We could see what alleles are there, right? What possible alleles can get passed on to the next generation. And so that lets us also calculate the genotype frequencies and allele frequencies. So genotype frequencies are basically the percentages of individuals possessing each genotype. And so if you look at the picture here, we've got some moths. And so they sampled, 500 moths. And of the 500, 320 of them were homozygous dominant. So out of the entire population that they looked at, 320 out of 500 were homozygous dominant. And so we've got a genotypic or genotype frequency of 0.64. 64% of the population is homozygous dominant. 32% is heterozygous, and 4% is homozygous recessive. So those genotype frequencies are just calculated by looking at what alleles are represented in your population and basically just dividing them up into proportions. We can do the same with allele frequencies. So with allele frequencies, it's very similar to genotype frequencies, except we're looking at the frequencies of individual alleles. So of the 320 individuals that are homozygous recessive, homozygous dominant, since each of them has 2 dominant A alleles, then that means that there's 640 dominant A alleles represented in the population, right? Of the heterozygotes, there's 160 of them, and so that means there's 160 of the dominant A's and there's 160 of the recessive A's. In the 20 individuals that are homozygous recessive, that means that for each of these individuals, there are 40 recessive versions of the allele. And then when you calculate this out, your allele frequencies would be, of all of the alleles present in this population, 0.8 or 80% of the alleles are the dominant version, and 0.2 or 20% of the alleles are the recessive version. We keep track of these with 2 letters. P keeps track of the frequency of the dominant allele. Q keeps track of the frequency of the recessive allele. So we would say P equals 0.8 and Q equals 0.2. All right, and we can calculate this too by using the equation P squared plus 2PQ plus Q squared. And hopefully you guys learned that in Dr. Whitney's portion of the semester, where that's this calculation is used um, to calculate Hardy-Weinberg principles and to do Mendelian genetics. All right, but we won't really be using it. Okay, so let's talk about null models and genetic equilibrium in Hardy-Weinberg. So first of all, null models. So a null model is a conceptual model that predicts what would happen if a particular factor has no effect. All right, and so let me just go on to Hardy-Weinberg. So the Hardy-Weinberg principle is a null model. And the Hardy-Weinberg principle is a null model because it predicts what we would see if a particular factor, and that factor is evolution, has no effect. So in other words, in Hardy-Weinberg, we're saying, okay, well, let's look at the allele frequencies in a population. And if it's not evolving, then we would expect the allele frequencies not to change, right? And if the allele frequencies are changing, then the population is evolving. And if the population is evolving, then it's violating Hardy-Weinberg. All right, so in other words, the Hardy-Weinberg principle assumes that there's genetic equilibrium. Genetic equilibrium is simply the point at which neither allele nor genotype frequencies change from generation to generation. So if a population is not evolving, then it would stay at genetic equilibrium. So we would look at, to go back here, we see that we can calculate our genotype frequencies in this population of moths, 0.64, 0.32, and 0.04, and then our allele frequencies are 0.8 and 0.2. So Hardy-Weinberg would say if this population is not evolving, we would expect the allele frequencies to stay the same over time, and the genotype frequencies. So we could come back 5 years later, look at the population, and if we calculate the genotype and allele frequencies again, we would expect them to be the same. They would remain the same if evolution isn't happening. However, if evolution is happening, then the genotype frequencies would change and the allele frequencies would change, and we would say this population is not in genetic equilibrium, and therefore Hardy-Weinberg is being violated and the population is evolving. So how would a population maintain genetic equilibrium? How would it not evolve? Well, for a population to not evolve, it basically has to meet these 5 aspects of the Hardy-Weinberg principle. So for a population not to evolve, there would have to be no mutations occurring, right? And that makes sense because, and we'll talk about more of these in more detail in part 2. If no mutations are occurring, then we're not generating new alleles, right? A mutation is going to change DNA, and it's a source of genetic variation. So if mutations are occurring, then we would expect new alleles to be popping up. And if new alleles are popping up, then they're going to change these genotype and these allele frequencies. So to remain in genetic equilibrium, the population can't have mutations. To not evolve, right? For the population to not be evolving, then it can't be having mutations. Number 2, the population has to be closed to migration. So for it not to evolve, there can't be new individuals migrating into the population. Why is that? That's because when an individual migrates in, it's going to bring new alleles with it. And when it mates with other members of the population, then that can affect the genotype and allele frequencies. The population is infinitely large. So in other words, for a population not to evolve, it has to be infinitely large. Any population smaller than infinity is going to evolve and violate Hardy-Weinberg, and that is a little more complicated. We'll come back to that one next time. Number 4, all genotypes survive and reproduce equally. So in other words, for these genotype and allele frequencies to remain the same, then it shouldn't matter in terms of survivorship if you're homozygous dominant, heterozygous, or homozygous recessive. All right, and so for the population not to be evolving, then it doesn't matter what genotypes they have. Number 5, individuals mate randomly with other members of the population. So for a population not to evolve, then random mating has to occur. The population would evolve if the having a homozygous being homozygous gives you an advantage in gaining more mates and having more offspring than being homozygous recessive, for example. All right, I don't know if that made sense, but that was the overview. Um, and again, in part two, we're going to come back and talk about each of these in more detail.